Okay, so good morning, everybody, and welcome to my talk today, which is about the use of the MGL in the public health context. So I work for the Public Health England Meningococcal Reference Unit. We're the national reference unit for Neisseria meningitidis for England, Wales and Northern Ireland. And the services we provide include species confirmation and meningococcal characterisation for the purpose of things like case contact management, outbreak investigations and surveillance, and we maintain an archive of currently approximately 100,000 isolates. And from an MGL perspective, of course, we also form part of this chain of laboratories that begins at the patient and ends at the MGL. So the local microbiology laboratory take the initial culture, and then this comes to us for DNA extraction, quantification, serogrouping, and also we gather the metadata. And then we send this off to a sequence provider who perform the sequencing, and then Martin's team at the Maiden Lab assemble the data and deposit it on the MGL, where it can be automatically and manually curated. And there's an army of curators out there, which I'm sure you're going to hear about later on. And so what I want to talk about today is sort of this feedback that we get from the MGL, how it feeds back into what we do in the reference unit. And I'm going to look specifically at characterization, surveillance and enhanced surveillance, then outbreak analysis, and then strain sharing and research. Okay, so the sort of core meningococcal typing targets, if you like, are the capsule, which gives us the group, two outer membrane porin proteins, POR B and POR A, which give us the type and the subtype. And we still look at these in the traditional way, using specific antibodies to characterize them. But also, of course, these days we can use PCR and sequencing. And whilst these are a little bit slower, for example, in an outbreak situation, they give us much greater level of resolution. And of course, using PCR and sequencing, we can also characterize these intracellular proteins or gene fragments thereof for multi-locus sequence typing. So obviously the capsule is incredibly important to characterize because it's the target of several major vaccines. So this is useful for case, contact and outbreak management, and also for directing vaccine policy. The two porins are traditionally useful for outbreak detection, and MLST, because these are sequestered away from the host immunity, they're not under any kind of immune selective pressures, and so these are particularly good for population biology. And together with antibiotic resistance testing, these form what we would regard as routine surveillance. But as many of you will know, in 2015, September, we introduced the meningococcal B vaccine, 4C-MenB or Bexsero, and as well as porin A, this includes three further outer membrane proteins, which are FHBP, NHBA, and NAD-A. And so now we need to characterize these as well, and we do this genotypically, but also phenotypically using this meningococcal antigen typing scheme. It's an ELISA, obviously because it's important to know how much these are expressed on the surface. So the reason we characterize these, of course, is for vaccine surveillance, to see how the vaccine might work if we were to use it, see how the vaccine is working if we are using it, but also for outbreak management, so that we know if a vaccine is likely to work in any given scenario. And this forms collectively what we call enhanced surveillance. And so you can see that in a short period of time, the number of targets that we characterize has increased quite considerably. The max is enough to keep one person busy for an entire year. And having moved from a sample of isolates each year for MLST to doing all of them, plus these three antigens, that's enough probably for 2 or 3 people as well. So the MGL couldn't really have come at a greater time, because to answer the question, what is the role of the MGL in characterization, surveillance and enhanced surveillance? Well, the answer is that the MGL provides all of our culture genotyping for the MRU. So it's really a massive step change. And of course, it's future-proof as well, because any new vaccines that come along are already covered. Okay, so now I'm going to move on to outbreak analysis. I'm going to be looking at national and international outbreaks first, and it won't be a surprise to many of you that I begin with the serogroup W ST11 outbreak in the UK that began in 2009. So you can see here, just about the time that the library was being established, we started to see this increase in group W disease due to ST11. And alarm bells began ringing immediately, because we were part of this large global outbreak of WCC11 back in the early 2000s that was associated with pilgrims returning from the Hajj. And it was associated with higher death rates and morbidity, so a particularly nasty strain. So we were concerned. And it's also an interesting strain, WCC11, because prior to the Hajj outbreak here, because it mainly caused sort of sporadic cases, small outbreaks, but then following the Hajj, a year or so later, we saw the first of several massive outbreaks in sub-Saharan Africa. And from the early 2000s as well, we started to see endemic disease increase in South America, South Africa, and now, of course, we're seeing it in the UK as well. So certainly we were concerned. Now, traditional typing schemes make WCC11 isolates all look identical to each other. They've all got the same pore A, P1.52, and they're all ST11. So previously, this strain in general has been known as the Hajj strain, and it's sort of widely believed that all of these later cases related back to this initial Hajj outbreak. But of course, in the days of the MGL and accessible genomes, we were able to look at this more closely. So with funding from Martin's Wellcome Trust grant, we were able to look at a comprehensive panel of CC11 isolates, 738 in total, from 25 countries across 6 continents and over 6 decades. So what would look like this using MLST? So you can see that the vast majority of isolates belong to this central sequence type, ST11. This, as you can see, there's much more structure here when we move from 7 loci to the 1546 core loci that we used in this case. We found that all of our group W isolates fell up here on the left-hand side. And if you imagine that the evolution of this takes place in this direction, you can see that they actually break off into 2 distinct sublineages. One on the left includes the strains associated with the global Hajj outbreak, and also the sub-Saharan African epidemic strains and the endemic disease strains from South Africa. And then on the other side, we see the South American strains and our endemic UK strain as well. And so this has subsequently become known as the South American strain sublineage, named after this South American strain. And similarly, this is the Hajj strain sublineage, named after the earlier Hajj outbreak. And knowing the experience of the South Americans, and this is a nasty strain as well, this prompted the introduction of MenACWY vaccine in the UK from August 2015. So this is a real world massive impact, in fact, that the MGL had from a relatively early moment. Okay, so now I'm going to move on to small focal outbreaks. So this is a paper relating to a protracted 4-month outbreak of invasive disease associated with a nursery. So you can see here that in the February of the corresponding year, we had 2 cases, 9 days apart, and these both had this very rare pore A, P1.18 for 2538.1. So as you'd expect, appropriate public health measures went in, and they went in chemoprophylaxed 2 classrooms out of 4. And that was that, or so we thought, until 18 weeks later, there was a 3rd case. And this was initially assumed to be distinct, not related to these, but then of course it came up with exactly the same incredibly rare pore A. So this was bound to be related. And so once again, chemoprophylaxis was applied, but this time it was applied to the whole nursery. And at this time, we actually went back and we did a retrospective analysis of pore A from all of the cases in the surrounding area. And we found another 2 cases with this pore A. One was a relative of a child at this initial nursery, and one was a child from a different nursery. So of course, you can see that the pore A has done an amazing job of highlighting this outbreak and allowing us to look at it. But so why do we need genomes? Well, the only reason the pore A really worked here is because it's so rare. So if this was one of the more common pore A's that accounts for perhaps 20% of isolates, there would have been a big question mark over whether case 3 was involved. And case 5 almost certainly wouldn't have been brought in either. So what did the genomes tell us? So there were 3 genomes from among these isolates. There were 41 CC4144, When we compared them to all of the CC41, 44 isolates on the PubMST and MGL, we fell into this cluster here. We did a separate analysis, and you can see that all three of these cases are really tightly linked to each other on here. So there's no doubt at all that these three cases are linked and part of the same outbreak. What's particularly interesting here is, although we know these are part of the same outbreak, they actually have different FHBPs. So FHBP is one of our typing targets that we use for enhanced surveillance, one of the BEX0 genes. You can see that this one's got a different one. And it's not unusual for meningococci to actually change the DNA, they swap it with each other. But during the course of an outbreak, it's quite rare for it to happen to one of our very few typing targets. And if this would have been POR-A, for example, then there's no way without this this high resolution genomic typing that this would have been included. Also, by going back and looking at all CC41, 44 cases, we would have identified any further cases associated with this outbreak as well. Of course, it's also incredibly useful for excluding small focal outbreaks. So we often get asked by health protection teams whether pairs of isolates, pairs of cases that are quite close in time and place, but with no apparent links, whether they are related. This is the South American strain sublineage again. We were asked about some cases at university. You can clearly see the two isolates fall into two different strains, South American strain, the original UK strain, so they were definitely not related. Similar story for this second university, two different strains once again, and then for this school as well, two different cases. So it's incredibly useful for, it's really a step change in looking at outbreaks. So what's the role of the genome sequencing and the MGL in outbreak analyses? Well, basically it's revolutionized outbreak analyses. So finally, I want to move on to strain sharing and research. So since the genome library's been established, obviously it provides more or less a catalogue of strains for people to go away, look at, do their research, and if they decide that they need to obtain some of those strains, they can come to us and sign a material transfer agreement, and as long as they're competent in using meningococci, then we'll send them over. This is an excerpt from a short talk I did back in the MRF conference in 2017 regarding the establishment of the MRF MGL as a keystone of global meningococcal research. So it was associated with a poster. It was a particularly large poster, as you can see here. This is actually to scale. And the reason it was so large is I wanted to go away, find all of the papers at this point that had cited use of the MGL. So I gave a sort of a short one-liner for each of them, put them into various different topics. And I wanted to include the reference list, a full reference list at the bottom, because that's what the poster was all about. So what did we find? Well, we found that the numbers of publications increased year on year, as you can see here. We seem to get this dip every other year, which I put down to not coinciding with the International Pathogenic Neisseria Conference. We identified 48 peer-reviewed publications in total, and these covered diverse topics such as population biology, epidemiology, outbreak analyses, and so on. And at the end of the talk, I gave an appeal to people to continue to cite the library, and I said that in a couple of years they might end up on a much, much bigger poster. So did this come true? Well, if you look at the following 2 years, and I say absolutely it did, because we've seen exactly the same increase in the number of publications. But then, of course, in 2020, we all know what happened. COVID came and disrupted everything. And yet, despite this, you know, I think there was still a fairly healthy number of publications in 2020 and 21 as well. So what's the role of the MGL in strain sharing and research? Well, after sharing hundreds of strains and at least 100 publications and counting, I think it's plain to see that it's one of the biggest, probably, charity-led interventions in recent years. I briefly want to look at the future and the inclusion of non-culture cases. So this is sort of almost 30 years of culture-confirmed disease from the MRU. Here you can see the strains that are involved in the MGL. But what this doesn't show is these PCR only confirmed cases. So we introduced PCR testing in 1996. And for these ones, we don't receive any cultures. We call them non-culture cases, and they account for almost half of all cases, and they occur because of early administration of antibiotics. So the bacteria are no longer alive. We can't culture them, but we can detect them using PCR. And in 2018, Steve Clark from our lab and Judy Brewer's lab found that you can actually remove the meningococcal DNA straight from blood samples and CSF samples using RNA baits, and that you can use this to create genomes. And currently work's ongoing to obtain non-cultured WGS for under 5-year-olds back to 2015, so we can start filling in this gap. The trouble is it's quite expensive, requires high amounts of DNA, so it's not something everyone can do at this time. Indeed, this project is funded by GSK. More recently, Sinbang's group from CDC showed that selective genome amplification is relatively cheap. It still requires high DNA, but certainly it's a step forward. So watch this space, and hopefully we can start to get closer to 100% of all cases in the MGL. And finally, I just want to look at what's going on post-COVID. So obviously, many of you will notice that we saw this massive decrease in meningococcal disease in the last couple of years, and this is because of the impact of social distance measures. And this asks a lot of questions. So, will the cases return? I think generally it's regarded as yes, they will return at some point. A more contentious question is, when will they return? Some people believe that sort of the waning immunity as carriage has gone away will lead to a large increase of cases at some point. Then again, earlier modelling, sort of early in the first lockdown, suggested that the impact on carriage at least might last for 5 years. So I think the jury's out on that one. But for me, the really interesting question is what's going to return. Is it going to be the same as we've got now, where, for example, we've got between 70 and 80% of MenB cases covered by a MenB vaccine, or perhaps a new strain will come along, or a new epidemiology that perhaps won't be so favourable. So on that note, I think you'll all agree that the need for surveillance in the MGL, despite this low number of cases, is probably greater than ever. And I will leave it there. Thank you very much. And thank you also to all of our collaborators from over the years. And apologies if I've missed anybody off.